Hi everyone, welcome back to this lecture on gene expression, focusing on protein translation. In the last part of this lecture, we went over the genetic code, the role of tRNA in translation, and the function of the ribosome in different stages of translation. In this part of the lecture, we will discuss how mutations in coding DNA can affect translated proteins. We come back to the central dogma, as illustrated in this figure showing transcription and translation. I can also illustrate this in a more general, simplified drawing, using block arrows as a common representation of the open reading frame or coding region of a gene. Showing the common elements of a gene, we have the promoter, which is where transcription initiates. Then we have the open reading frame, which encodes the protein, beginning with a start codon and ending with a stop codon. And we have the terminator, which is where transcription ends. The start and stop codons on mRNA and DNA define the open reading frame, but this is not the only sequence that is transcribed from DNA into RNA. I'd explained before that there is additional flanking sequence upstream and downstream of the coding region, called the 5' untranslated region and the 3' untranslated region respectively. We didn't really discuss the function of these sequences, but in the last part of the lecture, we saw that initiation requires the ribosome to bind at the start of a coding region, and the 5' untranslated region has an important function in ribosome binding. In prokaryotic mRNA, we have what is called the Shine-Delgarno sequence in the 5' untranslated region, just a few bases upstream of the start codon, and it functions to bind the ribosome. In eukaryotes, there is a special 5' cap on mRNA that binds ribosomes. We'll touch on this more in later topics covered in the course, but for now we are interested in the coding region or open reading frame in DNA and what happens to the translated protein as a result from mutations that change this DNA sequence. First of all, what is a mutation? Mutations are changes that occur in the DNA sequence. These can result in variation in living organisms and are a natural part of evolution. A mutation in DNA can result in a change in a protein, or it might not. Changing a nucleotide in a codon can result in a change in the corresponding amino acid. But it's also possible that a change in the nucleotide sequence may be a silent mutation, where the codon is changed to another that codes for the same amino acid as a result of the degeneracy of the genetic code. We know that many amino acids are coded by multiple codons. Shown in the figure on the right is an example of a point mutation where we have a substitution. The GC base pair is substituted by a TA base pair. Other mutations can involve insertion or deletion. Inversions are another kind of mutation in which sequences of bases become reversed or flipped. Focusing on substitution mutations that occur in coding regions, these could be characterized as silent, missense, or nonsense mutations. A silent mutation, as I explained, doesn't result in a change in the amino acid sequence, it just changes one codon for another, which codes for the same amino acid. A missense mutation changes the amino acid in the coded protein sequence, and a nonsense mutation results in a premature stop, essentially truncating the polypeptide that's being translated. Mutations can vary in the degree of their effect on protein function. Conservative mutations result in changes to the protein that have minimal effect on function. Some mutations can result in a significant change in protein function, and often mutations can impair or destroy protein function. An example from your textbook is sickle cell disease, or sickle cell anemia, which is an inherited blood disorder. It results from a mutation, a change in one base pair in the gene encoding the beta subunit of hemoglobin. This leads to a drastic change in the protein product, and the result is that the red blood cells that carry hemoglobin are abnormally formed and blood flow becomes restricted. The disease is inherited recessively, meaning it results from two alleles of the mutated gene encoding this protein, one from the mother and one from the father. So a homozygous genotype with two copies of the mutant gene causes sickle cell disease, but the mutant allele is often found prevalent where malaria is endemic, and the mutation may represent a potential evolutionary trade-off, since heterozygous carriers of one recessive allele don't have the disease even though they produce the mutant hemoglobin subunit along with the functional subunit. And these individuals are apparently more resistant to malaria. Thus, the mutation may confer an advantage in some conditions despite its otherwise negative effects. Looking more closely at how the sickle cell mutation affects the function of the hemoglobin protein, we see in this illustration that the mutant protein aggregates, whereas the normal hemoglobin does not. Red blood cells that carry the aggregated hemoglobin become misshapen, like a sickle. The cause of this effect can be traced to the change in a single amino acid near the N-terminus of the protein subunit. This change was identified in the 1950s using a technique that's described in your textbook called protein fingerprinting. This was carried out by digesting the protein and running gel electrophoresis. First, trypsin cleaves the protein into distinct peptide fragments, cutting specifically at lysine and arginine residues. Then, the peptides are separated in 2 dimensions to produce a fingerprint. In one dimension, the peptides are separated by size using electrophoresis, and in the other dimension, they are separated using chromatography. This will give a distinctive pattern for a protein that is treated this way. The fingerprinting could be applied to normal hemoglobin and to the mutant hemoglobin from sickle cell anemia patients. So that's what Ingram and his colleagues did in 1957, performing a protein fingerprinting procedure to chop up the hemoglobin protein, normal or mutant hemoglobin protein with a specific protease, and then to separate the peptides. Each of the peptides can be seen on these so-called fingerprints as distinct spots. Each spot corresponds to a different peptide fragment, and they saw that the fingerprints of the normal and mutant hemoglobin differed by 1 spot. You can spot the difference. This is the peptide that differs between mutant and wild-type protein. It was identified as an N-terminal peptide fragment on the beta subunit of hemoglobin. The amino acids in these peptides could be determined by peptide sequencing using Edman degradation. After isolating the peptide from the 1 spot that differs, they performed peptide sequencing and compared the N-terminal fragment from the normal hemoglobin, HbA, and the N-terminal fragment from the mutant hemoglobin, HbS. They determined that the mutant protein differed by only 1 amino acid at position 6 from the N-terminus, where the mutant had a valine in place of a glutamate compared to the wild type. As shown in this slide, this was the result of a point mutation at a single base in the nucleotide sequence. A single base substitution changes the codon for glutamate to 1 for valine. So we have seen here an example of how a mutation, changing the sequence of DNA, can have a specific effect on protein function. In Chapter 3, we've learned that gene expression usually involves 2 important processes, transcription and translation. Producing a protein that is encoded on DNA goes through these steps. I say usually because there are some cases where the final product that carries out the function of a gene is not a protein, but RNA. We've encountered some functional RNAs in our past lectures as examples. You might also think of some examples where protein synthesis may not involve transcription of genes encoded on DNA. In any case, we've seen in this chapter a brief overview of transcription, the DNA templated synthesis of RNA by an RNA polymerase. And we've also seen an overview of translation, in which the transcribed mRNA is decoded by the ribosome to synthesize proteins. That concludes our lecture wrapping up Chapter 3. You can find additional reading in the textbook and exercises in the problem set and textbook questions.